Hello everybody, welcome back to another lecture. Before we get started, if I could just grab a couple of thumbs up that you can hear me on Zoom. Yeah, perfect. Let's get on with it. I'm Nina Quinn, and today we're going to be using the Kaiwharawhara catchment in Wellington to discuss the relationship between E. coli levels and human influence in rivers. I'll just quickly introduce you to our lecturers today. There's myself, Sutherland, Felix, Silva, Wanping, and Sophie. We've come together as a team, to work on this particular project. But first, I'll be handing over to Felix to get us started today.
Thanks, Nina. Kia ora, everyone. I'm Felix, and as Nina mentioned, our research topic is focused on the human impacts on the Kaiwharawhara stream. To quantify this, we chose E. coli as our main indicator and also rubbish count. So this is the Kaiwharawhara catchment, and these were our sampling sites. 2 at Zealandia, 2 at Aotearoa, 2 at Jurassic Park, and 1 at the estuary. So why we chose this topic? Why do we care? We don't need to look overseas to see the impacts of E. coli in New Zealand. For the past few years, this issue is just becoming more common for New Zealanders. But how these line up with the literature? You can see on the screen some of the papers which refer to the impacts of E. coli on streams due to human actions. It is known that urban streams might have significantly higher levels of E. coli compared to streams dominated by native environments. And unfortunately, the Kaiwharawhara stream is no exception. The report on the left indicates that E. coli is one of the main sources of pollution coming from urban areas, more specifically from wastewater overflow after rainfall events. The paper on the right, however, explains how we can reverse past environmental damage and restore the model Kaiwharawhara stream as done by Zealandia. And that is our inspiration. Now I'll leave you with Wanping, who will introduce our hypothesis based on the literature. Thank you.
Thanks, Felix. So based on this background information, here in our study, we expect there is a strong relationship between the increase of E. coli levels downstream the Kaiwharawhara catchment and anthropogenic influence from Zealandia to the stream mouth, as measured by rubbish survey and stream habitat visual assessment.
Awesome. Now we're going to move on to our methods. So we've got videos of us explaining them as we work, starting with water sampling, rubbish and habitat assessment, and then moving on to E. coli processing in the lab. Okay, I've got a labelled sample bottle and I'm aiming for the deepest and fastest flowing part of the stream. First I've got to rinse it out a couple times before I take the sample. So I put it in upside down, tip it over underwater, and then tip that out, and I repeat it. And then the third time I'm keeping the water, and that's our sample. And then I hand it back to Felix over here.
Thank you.
Who takes the sample and gives me a new bottle, and we repeat the process until we've got 5 at each site.
So basically what we're doing here is we're setting out 30 metres from where they're taking the sample down to down along the stream edge, and then from there we are looking across the stream, the water, and then 3 metres away and counting the rubbish.
Also, at every site, we're doing a physical habitat visual assessment where we rank the site based on 8 guidelines set out by NIWA. And then, once I have 50 mils, you place it into this bit and then over the liquid waste. And we would have a plate, this is example 1, and take it off and put it face up on a plate, like that. It ends up like that after incubation. So for every plate that we have sampled, we've left it to incubate for 24 hours, and now we are simply looking down the microscope and counting how many colonies there are. For most of our samples, we've quartered them, and we're counting the colonies in a quarter. Okay, so now I've just finished counting this quarter. I got 1,198 colonies. Then we're going to take this number and we'll times it by 4 to get what we expect to be the colonies across the entire plate. Then we'll average that out with all 5 of our samples from one site. And then we can divide it by the amount of mils that we filtered through with using this plate. So this one was only 50 to get our colonies per mil.
Hi everyone, so I'm Sophie. I will be showing our findings in our investigation and what we have found, really concluded. So just skip that. After taking all of our samples and counting our E. coli and looking at our rubbish scores and our habitat assessments, we have come to the conclusion that generally as you go down the stream, the amount of E. coli gets worse and the water quality becomes worse because the human influence is greater. So left-hand graph here, The rubbish scores and the habitat scores are very strongly related because as the habitat scores increase, so does the rubbish score, which is also confirmed by an R-squared value of 0.88, which shows a very strong correlation. The graph on the right compares the E. coli counts to our rubbish scores. So we found that this relationship was best described by an exponential model, with E. coli counts dramatically increasing when our rubbish scores were decreasing. In other words, the higher the human influences, the higher the E. coli levels were. One thing to note here is that the high rubbish score means that there is less rubbish. So it actually means the environment is better if there is a high rubbish score, just because that's how the SMAC one works. So here on the graph, the very super high E. coli level here is the sample that was taken from the estuary, which is the very furthest downstream of the catchment with a very high human influence. But the two sites here with about a score of 40 in terms of the rubbish are the ones taken from Silva-Levio, which is the very, very top of the catchment with a very low human influence. As you can probably guess, looking at our data and our graphs, we've been able to accept our hypothesis and be able to conclude that there is a significant relationship between the increase of E. coli levels downstream the Kaiwharawhara's catchment as well as human influence.
So, as she just mentioned, our results found that there is a significant negative relationship between rubbish scores and E. coli counts. As we approach the lower catchment of the Kaiwharawhara stream from Zealandia to the estuary, with a decrease in the rubbish scores, which means more rubbishes and higher levels of human-induced contamination, we found an increasing trend of E. coli concentrations in the stream, implying poorer water quality. This relationship was commonly found in rivers and streams in New Zealand and is consistent with a study carried out in the same stream a decade ago. However, E. coli counts were slightly higher in the Aotearoa bush than in the Triassic Park, which was closer to the stream mouth. We have three possible speculations about this unusual result. Firstly, there was a heavy rainstorm the night before we collected our samples in the Aotearoa bush. Thus, there might be higher sewage overflow and resulting in higher E. coli levels. Secondly, we found the diaper and the dog poop bag in our sample sites in the Aotearoa bush that could lead to direct contamination in our samples and higher E. coli concentrations. Lastly, when preparing our plates for E. coli countings, we filtered different volumes of water for samples from the Aotearoa bush and the remaining sites, 100 ml and 50 ml respectively. This could lead to errors in our counting and thus affect our final results.
Hi guys, thanks for having me. Today I'll be critically analysing and mentioning improvements that we can make for our study for the future. If we were to repeat this scientific experiment again, we would ideally increase the number of samples taken to have a larger sample size, which would better model the relationship between the E. coli colonies and the rubbish count throughout the Kaiwharawhara catchment. Another criteria to add to decrease the variability of our data would ideally be to have the consistent environmental conditions for all our samples taken. As mentioned before about the heavy rainfall, it would have affected the trend of our data and influenced it. But as we know in the field of science, experiments don't always go to plan and nature is not always perfect. Therefore, if we had more resources and time, we would take more samples for the same conditions and time of day, so that we can accurately assess the relationship between E. coli colonies and the urbanised influence. Also mentioned, we had a couple of bumps in the road whilst we were in the lab, so we had to change things on the fly. We had to change the volume of water in the filter from 100 mils to 50 mils, because otherwise it was going to be too hard to count, so the issue would be easier to count. Though for next time, ideally, we would want all the samples to have the same volume of water throughout the filter. Additionally, we'd also try to have the same incubation period for each sample, so that the E. coli colonies would have the same time to develop in the incubator on plate. This occurred due to the time crunch that we could not control, so for next time we'd repeat this process again. We would make all the plates have the same incubation period.
Perfect, thanks Amber. If I could just get a round of applause for our lecturers today. Woo! Thank you. I'd just also like to take this chance to acknowledge some others that have helped us through this research project. We've got Sydney and Katie, our TAs, Mel, the lab supervisor, and thanks to Papatuanuku and Tane. Right, that's all for today. Thanks for joining us, everyone. I hope you enjoyed this topic, and I look forward to seeing you all next week. Lovely. Probably shouldn't have done that.